Okay, let's start it all over. Today we are going to talk about the third level of our subdivision of the research project, the design of the experiment, which, as I said, is the third level. And I'll emphasize again that the levels are not entirely separated from one another, but that you seamlessly pass from one to the next or to the previous, depending on the needs of the research project and the obstacles that you might find during the execution of the experiment. That is, for example, you might have devised an experimental approach which looks sound, looks good, and yet once you start doing the experiment you realize there is a problem in the approach and you have to come back and modify the approach and consequently the experiments. So you go back and forth until things work smoothly or fairly smoothly. Perfect smoothness in research doesn't happen. Alright, so the planning of the experiment, I'll stay here for a while because here, planning of the experiment, what is it first of all? It's what you do usually the day before running the experiment, usually in the evening, in the late afternoon of a day's work. When you're planning the experiment for the next day, why you do it late in the afternoon? Because the next day you want to start, be ready in the morning as soon as you arrive. If you have to design it, if you need to design the experiment in the morning, then you start in the afternoon, and you'll probably be late at night, or you won't even finish the experiment. So usually you plan an experiment beforehand. What do you need to plan an experiment? Well, many things. First of all, you ask questions. Do we have the right protocols? Oftentimes you run an experiment that you are familiar with, that the laboratory you work in has run many times, you use familiar techniques, so that's not an issue. But other times you are using a new technique or you're adapting previously used technique that nonetheless needs changes, and so you ask yourself, do we have the right protocols? Protocols are to research what recipes are to cooking. You can have a recipe in your mind and cook whatever according to the recipe, or you may have to read it because you don't remember it. And so you need to have it written somewhere. Same thing for the protocols. You might be so familiar with the technique that you can do it without looking at anything, or you might have the need for a protocol. Protocols are basically a series of steps you have to go through in order to apply a technique. They, nearly by definition, are never complete. There is no such a thing as a protocol that you can follow literally and come to a perfect result. It's like a recipe in the kitchen. You are not a great cook simply because you own a book of recipes. No, it's not sufficient to read a recipe and apply it. There are 1,000 things that cannot be written down, or that are too long, too tedious, too immaterial to be described in a protocol. And you have to learn them the hard way, that is by trial and error. So start with the best protocol and then figure out what you really need to do, how, as it is often the case, you have to adjust it to the conditions you are working with, you're working in. and then proceed. Once you can can answer yes to this question, we go to the next. Do we have the reagents we need? Usually the protocol specifies the reagents necessary to run the experiment. They can be anything from chemicals to antibodies. It could be not only reagents, of course, it could be pieces of equipment, instruments, whatever is necessary to do what the protocol specifies from beginning to end. And of course you have to verify in advance that you have all the reagents needed, otherwise you will stop in the course of the experiment. And so you go through this check. And it might happen that you have to postpone an experiment because while verifying this, you realize that one ingredient has been used up in the laboratory, is no longer there, or you never had that particular antibody and you need to buy to order it, or that something that you thought was there has expired, you cannot use it, etc., etc., etc. Then you ask, what are the execution times? What are the execution times, of course, for the experiment, for the protocol? How many hours? How many steps? How many incubation times during which there is little to do about the experiment and you can spend the time doing other things, etc. So, for example, a typical question is, will I be able to do this experiment all in one day, or will I have to stop at some point? And in that case, at what point? There are points at which you can stop and others at which you cannot, because that will ruin the experiment. Who will run the experiment? In a laboratory, somebody has to take responsibility for the experiment. Maybe the experiment, more people concur to the experiment, but there is usually a person who is the one responsible for that particular experiment, if not for the whole project. So it is necessary to identify that person because he will make the final decision as to what to do and what not to do, and because he has responsibility for the experiment, as I said. And also, of course, will be basically the head of the experiment itself. Then will it be necessary to work unusual hours? Sometimes it is necessary, despite all you do, it is necessary to come late at night or very, very early in the morning or a combination of the two. That's because biological materials, cells, animals, don't wait for you. They have their own times and you should follow them. So that should be planned in advance. Then, will there be any risks? Because of course in the laboratory we use chemicals, so there is a chemical risk. There is a physical risk, such as radiations, such as heat, such as extreme cold. There is a biological risk in case infectious agents are present, or even a biological risk associated with things you don't know whether or not they are contaminated by pathogenic agents. So you have to evaluate all this. And then, will the economic costs be sustainable? That is, okay, I can plan a perfect experiment, but it costs too much. The reagents necessary are too expensive. Or it would cost 1 billion cells. We don't have the room, we don't have the money necessary to grow up 1 billion cells, etc. And many, many other considerations. So I made it this very partial list, not to scare you, not to give you the impression that running an experiment requires a year of planning. No, it usually takes just an hour or so. But the thing is, there are many, many things that concur in the success or failure or the failure of an experiment. And you have to be careful in planning the whole experiment in all its phases. Generally, it is not that difficult. Generally, you know already what you need to do. It is sufficient that you plan what samples you want to have in the experiment, what are the positive and negative controls, verify that the experimental design is accurate. Then you know that everything is already present in the laboratory, you have a good protocol, you have probably used the same procedure before, you know it by heart, so many times you don't have any problems. When you do, or when you're planning on doing a new experiment, using a new technique, then it takes more thinking, it takes more time, it takes more planning. And it is rewarding, though, to invest that time in thinking and planning, because then the experiment works. On the other hand, one opposite consideration is that at times some people who are very accurate, very painstaking, might spend too much time in planning an experiment. So trying to think it through very carefully, very thoroughly, very completely, and never actually doing the experiment. If you're investing too much time in planning the experiment, then it is better to run a first experiment in a reasonable way, then rather than trying to anticipate the million problems you might encounter, just ask, after I've done the first experiment, how many problems have I found? 2. Okay, I'll face those 2, I'll solve those 2 problems and forget about the others because they simply didn't show up. So it's a matter of having some experience in planning the experiments, but I can assure you that it is not as difficult as this list would suggest. So, as I mentioned, other than considering the techniques themselves involved in the experiment, you have to design carefully the experiment in order to isolate properly the variable at study and introduce the appropriate controls. For example, let's make an example and consider some aspects of it. We wouldn't have the time to go through all the aspects of such an experiment, especially because you don't know, you don't have experience, you don't have familiarity with the techniques involved. But we'll consider some aspects to give you a sense of what the actual planning of an experiment is. So let's suppose that the purpose of the experiment is to verify the transcriptional activity of this transcription factor, which I think we have mentioned before, in our experimental system. That is, we know this is a transcription factor, we know hence that it has transcriptional activity, but I would like to know whether it has a transcriptional activity in my system, the specific system, because it isn't necessarily active everywhere. Perhaps in my system, a necessary cofactor does not exist, or there are inhibitors of the transcription factor, or some other conditions are not met. So I want to ask whether in my cell type of interest, this transcription factor works. And so this is the first question for you today. How would you measure, how would you assess whether the transcription factor works in a specific cell line? Do you know a technique to address this point?
Real-time PCR.
Real-time PCR, and you would amplify what?
I don't know if I have a gene associated to.
So you would amplify a target, a known target of the transcription factor.
Yeah.
That would be one possibility.
Since it is a transcription factor, we could do transform our cell line with the promoter whom this factor binds, and a GFP or another fluorescent marker, and measuring the light.
That's another possible approach. Any other ideas? No.
Maybe even an RNA-seq for having no, but that no.
It could be done, but it's very expensive and not necessary in this case.
No.
Not very expensive. Expensive. This is whatever we are going to do next is far cheaper, but in principle it could be used. Someone?
I don't know. Transcriptomics will be expensive.
This is what
Yeah,
just Daniel said.
Immunoprecipitation of our
Precipitation of what?
of the of what should the factor make. No.
So you would look for the protein product
Yeah.
of the gene
Of the gene
activated
by the
transcription.
factor.
Well, that's pretty cumbersome, and it involves biologically, it involves several steps. So if you don't find the end product, the protein,
Yeah.
you wouldn't know whether
If
where
yeah.
the process failed.
Maybe an immunoprecipitation of the transcription factor binded to the DNA.
That's called a chromatin immunoprecipitation, or rather a chip-seq. That is, you precipitate the transcription factor and determine what DNA sequences it is attached to.
And you get a qualitative data rather than a quantitative. I mean, you don't know the actual activity if you don't know how many
Exactly.
DNA.
It tells you that the transcription factor binds, but not necessarily that it is active. So it is a valid technique, but not for this purpose. And finally, Valeria, any suggestions? If not, we'll take a no for an answer for the time being.
Create a mutant cell for the promoter.
Oh, okay, yeah. So you would introduce a mutation in the promoter, but there are probably several or many or very many promoters bound by the transcription factor. So you would have an unknown number of targets. So, okay, we'll consider a range of possibilities, most of which would be applicable, but probably the most expedient, the cheaper and fastest among these is Daniela's first suggestion, that is using, say, the luciferase reporter attached to the binding site for the transcription factor. And so the experiment would look something like this. We would co-transduce, say, a plate of cells with a plasmid encoding the transcription factor and a reporter construct having one or more transcription factor E2F binding sites and luciferase as a reporter. Okay, so we do this experiment, which might look something like this. here we have the reporter, E2F binding sites plus luciferase. We added to both of our two initial cell culture dishes. And then to just one of them, we also add the E2F1 vector. Not the other. This is obviously our negative control. We don't expect much luciferase activity to be found here. And if we design the experiment this way and perform it, we find that indeed we have several orders of magnitude increase in luciferase activity when the transcription factor is present, which would strongly suggest that the transcription factor indeed is active in these cells. But then you already know enough to know that the experiment is not complete. Why is it not?
We should do another control with only the vector.
Yes.
But I have a question for a slide before. I was wondering if we are verifying the transcriptional activity of this factor in this cell type, shouldn't the cell type already produce the transcription factor? Or
It
why
might.
are we?
It might,
It might.
but
Alright.
not necessarily. Transcription factors sometimes, oftentimes, are active even in cells that do not produce them. But if you introduce them into the cells, they will induce transcription. So, but you are right. There might be what we call a biological background, that is, the activity of the endogenous transcription factor, if that was the question.
Sorry.
No problem. So, what Daniela said is correct. What else is missing, if any?
Positive
A
control.
positive control. We have a negative but not a positive control. Okay? And what kind of positive control would you envision?
If we know already some cells that the transcription factor are expressed is active in, and how much we can see there the luciferase and to have a quantification maybe.
Okay. Then a subtler question. Would that be a technical or biological positive control? Think about it. It is not immediate.
It would be technical because it's about the luciferase's activity and
Correct.
not okay. But we should need maybe to that technical control, I don't know.
We will probably need a technical positive control, but we also need a biological positive control. Or perhaps, even if we don't need it, as an exercise, we'll think about it. What might it be?
The positive control,
Biological.
the cells without no,
Positive control.
yeah.
Difficult, huh?
Yeah.
Any ideas?
The vector in other cells.
But again, it would
Yeah.
be a technical positive control because we're using
Yeah.
different cells.
Only the luciferase with another promoter
Yes,
for
in a way, I would call it again a technical positive control because it would ascertain that luciferase is expressed in appropriate
Endogenous
conditions.
factors that we know.
That's, I think, what Daniela was suggesting.
kinda.
Okay, so let me understand.
I was thinking more about the luciferase background I mean, we know that the luciferase works, but we don't know if it is activated by E2F. That's the question we want to answer. So we might express luciferase without E2F and see if the level is the same. I don't know.
Yeah, that's correct, but again, it's a technical positive control. And I was wondering whether we can design a biological positive control.
I was thinking about an constitutive gene and mark it with a luciferase. And we should know the efficacy of the transcription.
You would know that transcription exists in that cell, which
Yeah, should be.
otherwise the cell wouldn't be alive. Right.
Okay.
So basically, in these conditions, it is hard to think of a positive control, a biologically positive control. So we'll consider only the negative controls. As we mentioned before, it is not always possible to design a meaningful positive control. If you want to control our technique, yes, we can add positive control, technical positive control. If we want to verify the biology of the system, we'll have to wait until we get the first results, and then we can use perhaps them as a basis for positive control. So going back to the negatives, actually, yeah, Daniele suggested basically another negative control, which controls for a different condition. This one here controls for the fact that the transcription, the luciferase we measure is due to E2F and not to other factors in the cell. What Daniele suggested was to verify that it is indeed due to E2F itself and not the vector. So he said, let's have the vector alone without E2F and make sure that it is at least as negative as this one. And yes, this is indeed the empty vector. But then these experiments usually include many negative controls, such as these. Basically, a repetition of these same conditions, but this time without the reporter. Why would you do that? Well, as we said, this control here verifies that there is no that the vector itself doesn't produce luciferase activity. Here, we are not adding instead the reporter vector, but a version without binding site. So we have luciferase, but with no promoter. And that shouldn't be prescribed. Certainly it shouldn't be prescribed by E2F1 because we don't have an E2F1 binding site. And so, as you see, basically all of these negative controls work properly. They have very low basal activity, which might be due to a background endogenous E2F. And so we can conclude in the presence of the appropriate negative control that indeed E2F works in these conditions, in these cells. So this is just to illustrate once again the need to think an experiment in its components and think how you can best isolate a single variable. So altogether here we are making sure that this transcription and the ensuing luciferase activity is based on E2F, E2F binding, not the vector, and no other activity that might stimulate luciferase. So it's a variety of possibilities that we're ruling out one by designing appropriate negative controls. And as you can see, this experiment has 5 negative controls and a single in a single test. It happens. It's frequent in these kind of experiments. By the way, among other things, this is also a technical negative control. It is technical in the sense it makes sure that we are measuring luciferase when luciferase is present. Here we shouldn't have any luciferase, and indeed we are measuring only background levels of light. Let's think about an entirely different experiment. Again, these are simple experiments conceptually, but I want to illustrate the difficulties that you will face the first time you run such an experiment. The many things you have to think about. And once again, not because I want to say that I want to convey the idea that doing experiments is very, very difficult, but just that you have to think carefully about your experiment and consider all the variables that might impinge on its results. So, let's say at this time our purpose is to find the duplication time of a cell culture. The duplication time of a cell culture is of course what?
How many time it takes to duplicate a cell?
How much time it takes to double the number of cells in the culture, yes. Which, not stated, but it includes a set of conditions. The cells should be far spaced enough that they have room to proliferate. They should not be too sparse, because it is known that because cells feed one another, talk to one another, if they are too sparse, they grow very slowly. They should have plenty of growth medium in order not to starve for nutrients or growth factors, etc., etc. So we assume, though, that the conditions are perfect for the cells to proliferate as fast as they can. And so now we consider how do we measure the duplication time of a cell culture.
From the culture, at different time we piastriamo, come si dice in inglese?
Plate.
We plate at different time, and then we count how much at that time is the CFU in our culture, and then we see mathematically when it doubles in what amount of time.
Okay, what you said is correct. There is a simpler way to do it.
Okay.
What you said is one way to do it. Other ideas. So basically, what Samuele was saying, if I understood correctly, is that you should prepare a set of plates, count the cells at different times.
And there will be a correlation probably
With time, yes.
with time, and
Would
so we
increase
can go back to the time that it doubles, like have a math relation between time and CF2 we count.
Yeah.
We have only plate or liquid culture also.
In this case, we work with adherent cells, and so we have plates.
Okay.
Most of the adherent cells do not grow in suspension, so we wouldn't have a liquid. The growth medium is liquid, but you need to have the cells attached in order for them to grow. Any other ideas? Okay, so let's say Samuele goes to the lab chief and says, I have thought of doing this experiment in this and this way. And the lab chief says, well, you know, it's okay, but you can do it with less expensive labor. He simply says, okay, plate a given number of cells, and tomorrow count these cells. So the first day you plate 100,000 cells, and then you go back the next day at the same time. So time 2 is 24 hours. And you count the cells again. Clearly they are increased in number. You count them, and you find that they are 240,000. So Samuele goes back to the lab chief and says, what did you tell me to do? Now I have overgrown my cells. How can I determine my application time?
Yeah, that's the thing. Just the math correlation, you can still find the math correlation about it.
Correct. So there is a formula which looks ugly, but it's very simple in fact. Here it is. Duplication time equals time 2 minus time 1 times logarithm base 10 of 2, this is a fixed number, over logarithm base 10 of n2 over n1. Basically, what are these numbers? T2 is 24, the hours. T1 is 0. So this thing here is 24. Log 2 is 1 point or something. And this n2 over n1 is of course 240,000 over 100,000 and logarithm of this ratio. So it's very simple math. And you do the calculations and it comes out that the duplication time is 19 hours. So Samuele breathes, he hasn't wasted his time, goes back to the lab chief and says, okay, now you have your answer, the duplication time is 19 hours, but the lab chief says, well, you know, it's nice to know, approximately, but for our purposes we need really to determine carefully, accurately, the duplication time. Are you sure it's 19 or 20 or 18? How would you estimate the accuracy of your determination, your measure?
Do it in more plates and then statistics and so we have
By now you are masters of statistics, so the answer is simply to set, for example, 3 plates, count them, you get slightly different numbers in each case than the usual statistics, perhaps a student's t-test, and you'll find that the application time is 19 hours plus or minus some minutes. Oh, and the way to do it is to simply substitute the individual number of cells with the averages of the 3 plates. Okay, so that was a good start. A more difficult experiment now, because the lab chief is not happy with the application time alone. He also wants to, oh, this is what the application, not the application, the growth code looks like. It is initially, if you plate few enough cells, it will be initially slow because they are not dense enough, as I mentioned. Then you will have a linear part of the curve, provided this is a logarithmic axis. And then it will plateau simply because there is no further room for the cells to grow, or the growth medium is exhausted, or other reasons. All right. So the lab chief now says, good, Samuele, you've done a good job. So let's tackle another question now, which we are also interested in. That is, in these particular cells, how long does mitosis take? How long does it last? What's the time interval between the beginning of the mitosis and final cell division? at her side, Samuele doesn't know what to say. So the lab chief will ask him, well, but grossly speaking, what's your estimate? How long do you think a normal, a typical mitosis takes?
I don't know. The cell cycle has different phase.
Uh-huh.
Mitosis is the last, no,
The smallest?
the smallest. If it's 19 hours, I'd say maybe 1 hour or 2.
Okay. Daniele,
Half an hour, 1 hour.
half an hour, 1 hour. Why are you saying so? You remember this
No.
notion, or you made an estimate in some way?
I mean, an estimate based on really long forgotten.
Okay, you don't know where it comes from, but you have this number in your head.
I feel like it might be. It is?
Okay. Okay. Not the foggiest idea.
No, I think 2 hours.
Based on what?
I don't
You have
know.
to justify.
Yeah, I don't know. From what I know that it's not too long process, but I cannot estimate. I don't know.
I think you can. Let's see if Valeria can help you.
I think like 2 hours.
Based on what? You have to justify your guesses.
My knowledge in cells.
Because I know. It's lower because I know.
In the first year of biologia cellulare,
So like,
we studied mitosis and meiosis, and I think I remember it's like 2 hours because I thought it's long.
all right. So my own way of coming to an estimate would be this. Think of, no, I don't think I have, no, I don't think a slide. I have a slide. Think of a typical representation of the cell cycle. As Daniele mentioned, there are 4 phases, and you probably have a visual sense of what is the longest phase.
Duplication,
Duplication meaning duplication of what?
DNA.
DNA replication.
DNA.
Is it? Do you agree with that? No, Valeria doesn't. Valeria shook her head.
No, I think it's when the doubled DNA chromosomes have to separate themselves.
That would be mitosis, part of mitosis, right?
Yes.
But I was talking of the whole cell cycle.
Okay. I thought of the mitosis.
So what is the longest phase in the cell cycle?
Mitosis.
Mitosis era quella
That's not what you said.
No,
You
no.
said that the mitosis
It's,
goes from half an hour to 2 hours,
It's
according to
the
your
growth.
guesses.
When the cell grows.
Sorry, I didn't get the beginning.
The growth of the cell.
Okay, what do we call that phase?
G.
G1.
G1. G1. That's the longest one. Yes, it is, usually. Then, what's next?
G2.
It is a trick question. It is S phase. No? S is
Daniele
separation.
guesses S phase.
S is for separation.
Why are you saying that? Because you remember the schematic of the cell cycle, or because...
Because my girlfriend told me about this really a lot of times, but
Okay, that's a source. His girlfriend is the source. She
I
knew
agree.
something and she told him.
I really hope
It's
I
a better
listen
source
to
than
her.
us. So
really hope I listen and her more than I
You
actually
should
did.
start listening.
Okay, so...
we trust Daniel's girlfriend.
I don't know if we trust her. What do you
No,
think?
I don't.
No.
I wouldn't. Seriously, wouldn't recommend.
I think it was like the cell knows that it's going to start the mitosis, like to duplicate, so it has to grow even more. So it's like another growth period.
Okay, what do we call that growth period? If it isn't J1, it isn't S phase DNA duplication.
Growth 2.
G2.
Okay.
That's right.
Wait, there is.
And then it starts duplicating the DNA.
No,
No.
no.
That's on G2.
The succession is G1, S, G2,
Oh, okay.
M, which stands for mitosis.
S was the last. No, was. Now I'm trying to remember the image. Now that I have the order, because I had the image, I knew that mitosis was the last that was the lead.
The smallest.
Yeah, the
No,
smaller.
G1 is the largest.
Okay, and then we have two other phases. That is
Yeah, that are pretty much.
And G2.
Are pretty much the same.
toss a coin and decide. We have just two phases.
Cioè dobbiamo dire di queste due qual è la più lunga.
Uh-huh.
Growth 2.
Daniele finally has figured out the schematic of the cell
Somewhere.
cycle. So what's the longest? The longer of these
Yeah,
two
but,
phases?
It's like when avere sulla punta della lingua una parola, ecco qua ho sulla punta della lingua un'immagine, cioè che sta là ma non.
Can any of you have
La S è più.
I think G2. Growth is always
Valeria says
the
G2.
That's Valeria.
Yeah. 2 is bigger than 1.
Karim?
I don't really know.
Okay. But we'll say anything. So I'll leave the question open for the moment, and reason like this: if G1 is the longest phase, minimally, how long should it be? Since the whole cell cycle takes 90
More than a
hours.
quarter.
More than a quarter, for sure. We also know that M phase is very short, you
Yeah.
said that. So it
I'd
is
say half.
Sorry?
I'd say half of the time, maybe.
Roughly half.
Yeah.
So, okay. Then we have three more phases. One is M, then we have S and G2. And again, think of this schematic.
So maybe the other two will be like almost
40%, 10%, 40%, 10%. Something like this.
Okay,
G1S, G2, and
okay.
roughly.
All right, so according to Daniele, we have roughly 40% of the time, which is 7 point something hours in G1, more or less as much in G2, 10% in S phase and 10% in M phase. But Daniele is not terribly certain of what he just said.
I just remember another image.
Different from the previous one.
Yeah, it was the cell sorting. Because there
Excellent.
were
I
two
didn't
big-
mention it because many of you have never seen a cell cycle analysis with the cytometer, but that's what you might refer to.
Yeah, but there were 2 peaks,
Uh-huh.
and the biggest was it was the biggest peak because there were more cells in that phase.
Uh-huh.
I really wish I read more carefully the thesis. Jesus.
Okay, what Daniele is trying to remember is this sort of diagram, which is actually a cell cycle analysis at the cyclophoreator, which goes more or less like this. So on the x-axis you have DNA amount on an arbitrary scale. So if this is, say, 10. This is the diploid content before cells duplicate their DNA. Then the cytometer sees together, pool together G2 and M because they have the same content. So here, approximately will be 20 because the amount of DNA is twice as much as in G1. And then you have S phase. Modify a little the drawing. This is so,
Okay.
and what's between the two main peaks is S phase, that is an amount of DNA which is between 2n and 4n, right? The same goes for plants, so it is no excuse that you study mainly plants. So, according to this diagram,
But
where is time here? Oops.
yeah,
No time.
exactly.
That I read the question on your face.
It's the
But time is
epsilon
not-
axis,
No,
Time.
that's just number of
number of
cells.
cells.
Oops.
But number of cells kind of tells us the time because
Exactly, implies time.
implies time. So the highest number of cells, it means they stay more time there.
Exactly.
So we kind of have the time of
Mhm.
it. And if the height is proportionate to the reality,
It is.
okay, the G phase is like 3 times longer than the S and G2.
Well, you have to think of not of the height itself, but of the
Ah.
area.
There,
below the
okay.
curve, which, let's say,
Which is,
imagine that this is S, like this, S phase. This is G1, and this is G2 plus M. So if you think of the area, this is not very,
it's comparable, yeah.
yeah,
So it would be like 45, 40 and 5%.
something like that, yes. Okay, so, of course, we have done it in a setting that doesn't allow you time enough to think and to reason. But Daniele mentioned, since you don't remember a schematic of the cell cycle, it's kind of strange to tell you the truth. The cell cycle is everywhere. Biology is a huge cell cycle. In
Maybe we don't put this on.
Let's transfer this.
Il secondo semestre seguiamo il corso sulla mitosi.
the defining feature of biology is growth. Whatever doesn't grow is not a biological object. So anyway, we have come to conclude that the second largest phase, longest phase,
Is
is S phase, and then there is a situation which we cannot
Tell by the
tell
peaks,
here because
yeah.
we have pulled together
Yeah.
two phases, but clearly G2 and M together are not very long. So if we think of this, we might say this is perhaps like 50-60%, another 40% is here.
Yeah.
So the final G2 plus M is about 10-15% of the whole cell cycle time. And so let's divide it straight by 2. If
Yeah.
even if it is 15%,
7%.
7%, okay, 19 hours, how much is 7%?
If it's 20 hour, that's easier. It's 10% is. Oh my god, hour, minutes, I don't.
Never mind, say decimal error.
Decimal. The
Something like 1.5 hours.
yeah.
Never mind the details because this is just a rough estimate in the Fermi way of thinking. Okay, so we have come to the conclusion that G2 plus M is about 1.5 hours, so M can be more long than so much, longer than so much. But then we want to measure it. How would you measure time length of anything in this condition, in a cell culture? You have just used the proper reasoning without realizing it. When you said, yes, that graph implies time,
Okay.
you can apply that reasoning here, provided you do the proper thing.
Maybe if we think it like
First of all, you should determine how many cells are in mitosis in this range, I suppose,
Yeah.
right? How do we do that?
by counting the colony, and if we know now
No, you cannot count colonies because you want to know instantly how many cells are in mitosis at this time. So you cannot replay them,
Okay.
wait them for them to grow and count colonies.
A photo with a microscope.
Photograph. So you want to assess how many cells are in mitosis based on morphological evaluation. Yeah,
I think it may.
that's okay. Yeah.
Not counting 100,000 cells, but you count some of them and then you.
Yeah. For example, you might count mitotic cells in some microscopic fields, chosen at random on the plate so that you have a good sample of the plate. Yeah. Or if we don't trust, say we are inexperienced, it is possible to determine mitosis based on morphology. But say we are inexperienced, what can we do to more objectively and easily determine whether a cell is in mitosis or not? Karim is anxiously waiting for you because he doesn't have an answer, so he was looking at you.
I was thinking about colorant, like
Stain,
PGT,
Like
PGT we use for bacteria.
BGT, no.
But I don't know for-
Yeah, there are stains that you can use, yes.
I was thinking a method of selection, like trying to kill the cells that are not.
That are not mitotic.
Yeah,
In principle, you can do something like
like
that.
because with the bacteria, like penicillin doesn't kill the dividing cell, but kills the cells that are growing actively.
Yeah.
So I was thinking something like that, but I don't know if we can do with this cell type.
There is a technique that does approximately what you're saying. It is called mitotic shaking. You just shake the plate physically, and the mitotic cells will detach selectively because they are loosely attached when dividing. But I wouldn't choose that technique to accurately determine the length of mitosis.
The lab chief will not be.
He would.
Can I measure also the area of the cell?
You can, but how does that help?
I know that when they are dividing, maybe I can see an increase of the area.
That would be extremely complex. It is feasible but very complex. We're talking about something easy. Who said that we might stain the cells?
Yeah.
You do, okay. So, for example, you don't know this. You might use an antibody that recognizes a specific phosphorylation on serine 10 of histone H3. That's a marker of mitosis. So If you do that, you find a few cells stained as being mitotic. So now you can count these cells. You can also count the total number of cells in the plate.
Then follow them for a long time and measure it.
Now you can follow them because you have stained them, so you have killed them.
Okay.
We have to do more play now. We have to do with different planes and have a math. It's like a differential thing to know how.
So you want to know how many mitotic cells are at this time, then
Yeah,
1
and then
hour
time.
later,
Yeah,
2 hours later.
yeah.
Do
And
you expect 2 hours later there will be more or less mitotic cells in the plate?
that
Depends.
More.
More.
cells.
Why more?
But they expect that in 2 hours maybe some other cell went mitotic.
Okay, and the other, and those that were in mitosis at time 0 stayed mitotic?
No, you say that we killed them, so. But I think that they will say remain sane. I don't think they will complete the mitosis. So.
Or perhaps they did. Some of them certainly will have. Say the ones that are in mitosis, but at the very end of mitosis, 2 hours later, they will certainly have completed. So the question is, do you expect that the proportion of cells in mitosis will be different at different times?
yeah.
You do.
No.
Why?
It depends because we don't know at what point of the cell cycle we played it. So if we played it at a random point,
We played them while they were growing exponentially. That is, the distribution of the cell cycle was that shown on the blackboard.
okay, so yeah, it will be proportionate and not change in
The
time.
proportion will not change.
Will
with
remain,
time.
yeah.
There will always be the same proportion of cells in any phase at any given time. So we don't need many places. We just need
Which
one.
is if we know that.
And so we have the numbers here. We count 4 cells in mitosis, 100 not in mitosis.
How does this correlate time? It's not like 4 divided.
Daniele perhaps has an idea.
It may not be too much ethical, but you can have an internship.
You want to torture the cells.
No, but you can have an
Make them
internship
confess.
student. You can record a whole night and focus on some cells, and have an internship student to watch the whole video and measure how many times it needs.
It can be done that way. And
But
it
you
is
are
done
the
that way.
internship student in
But
this.
since you are that student, you don't want to use that method, which is very tempting.
I was assuming I was the professor.
Not yet.
Does the proportion of cell correlate linearly to the time?
Okay, what we have said basically is a certain percentage of cells, let's say 4%.
Yeah.
For sure, is always in mitosis at any given time.
So it's the 4% of the 19 hour.
19 hours, exactly. So here is the answer, very easy. We know that the application time of this culture is 19 hours. If during exponential growth 4% of the cells are in mitosis at any given time, then the mean duration of mitosis is 19 times 60, just to measure minutes rather than hours. Divided by 4% equals 45, 46 minutes. Right? Easy.
So less than 1 hour.
Less than, yeah. Typically mitosis takes between half an hour and an hour.
Avevi detto?
Senza dottorare nessuno
Questo è un
studente.
almanacco.
was his girlfriend.
Telepathically.
All right, so we have already measured two variables, but now the lab chief asks, what's the duration of S phase? Okay, how do we do that? Yeah?
Now that we know, like, that 4% of that area is occupied by cells in mitosis, can't we? But that's just to estimate, okay? No,
That's an estimate. You can actually measure it fairly accurately, but you don't have a cytofluorimeter in your lab, so you cannot use that.
We can stain the cell in this phase,
How
and
do you
then
do that?
by staining something that
We have
happens
already-
only in this phase.
We have already described the technique to stain, to mark cells in this phase. We can mark them by
The API.
making them incorporate a nucleotide analog called, for example, bromodeoxyurea. So, first of all, this is a very simple schematic of the cell side. So we want to measure the duration of phase. We label the cells with UDR. And the next question should have been, that you've seen the answer, where we should label them for a long or a short time with UDR. You see the answer?
I think short.
The answer is a short time because we want to mimic the situation we had with mitosis. We want to know at a given instant how many cells are in S phase. So if we label them for a long time, we give time for some cells that were in G1 to incorporate the label, to enter S phase and incorporate the label. And at the same time, we'll have cells that were initially in S phase that have gone through other phases.
But
So
we
will
see them
overestimate.
as
No, because they sum up.
Continuous.
yeah, we saw the G2 that whereas SS.
Yeah. So we would have G2,
Yeah.
the length of G2 plus M plus
Yeah.
perhaps a piece of G1. The more we label the cells, the more cells will be labeled. Until eventually, if you label them for twice as long as the cell cycle, all of them will be labeled.
But how do you know you didn't label them too short?
What would too short be? Why would it be too short?
Maybe a cell in the which is replicating the last hundreds of bases doesn't have the time to incorporate the BRDU.
Correct. So we will slightly underestimate the number of cells in a phase, but we can ignore it. It's negligible. It's a small amount. Very small.
And the lab chief is okay with that.
Yeah. All methods have a certain degree of accuracy, no more than that.
And then count and same as before.
Exactly. So we want to have a short incubation with UDR, just the time needed by the cells to incorporate enough UDR that we can see it, that we can recognize it with an antibody and see it by immunofluorescence. No more than that, otherwise we would label a larger number of cells that are actually in a space at any given time. So assuming this short incubation, this is another approximation. Assuming this short incubation is basically an instant, it will take about 10-15 minutes before we have a decent signal. So we are assuming that this length of time is negligibly long compared to the length of the whole S phase, then all we have to do is to determine how many cells are labeled. We come up with 40%, and so we conclude that the length of a phase is 40% of 19 hours, which is 7.6. And typically, indeed, the length of a phase is between 6 and 12 hours.
How long would the short incubation be?
15 minutes,
15 minutes.
10, 15 minutes. With a shorter incubation, many cells wouldn't incorporate enough UDR to be detected.
Do we need a technical control to assess this, or we just?
Yeah, we do.
Ah.
It's in the next slide. Here it is. This is another detail in the experiment. Because what we have seen in the previous one is basically the approach. We haven't really played with details. We have just said, okay, if we make a short incubation time, label the cells which have incorporated UDR, count them, we'll have the measure we need. Fine. But then you have to do the experiment and you have to think of the details. One among many is the exact duration of the incubation with UDR. Because if you label the cells for a long time, most cells will have a strong fluorescence for UDR. These are the nuclei of some of these cells that have been labeled with UDR. The dark ones haven't incorporated UDR. But then we need a short incubation time. So if you have never done this thing before, in advance you will have to find experimentally, empirically, how long the incubation should be in order to detect the signal. And then you'll find that perhaps 15 minutes is long enough to detect a signal, and you will label the cells for 15 minutes. How do you determine that this is long enough? How do you know that you are missing cells that would have been labeled with a longer pulse of UDR? It's in the bottom part of the slide.
But with no incubation time, what no incubation means no.
I'm asking you. What does no incubation mean in an experiment?
No BRD.
Time.
No UDR?
No, yeah,
Yes.
okay.
But the anti
The antibody, yes, we use the antibody, exactly. So, what is this? What do we call it?
negative.
Negative control.
Exactly. So this is a negative control, and yet you can see at least one slightly weakly positive nucleus, perhaps a second one that you probably can't see here. A few nuclei do have a slight amount of fluorescence. What do we call it?
Background.
Background. What is it due to? It is due to the fact that some antibodies attach non-specifically, and so you have a slight amount of fluorescence even if you don't incubate at all with UDR. So you know for sure that that is background. So,
But does it is enough to assess that 15 minutes are the time that?
not exactly, not exactly. But it will tell you which of these cells of these
Yeah.
nuclei
We can
you count.
subtract
You won't
the
count
noise.
this nucleus here because it's approximately as strong as this one. You know this is false. So you count this as background. You don't count. But you count these because they are clearly stronger than the background. And so if you label the cells, say, for 10, 15, 20 minutes, and the percent, the number of
Ah,
labeled
it's
cells
very
doesn't
big, yeah.
increase, it means 15 is okay, or even 10 might be okay. If you label them for 10 minutes and you get a certain amount, you label them for 15 minutes and you get a higher number of cells, 20 minutes an even higher number of cells, then it means the shorter incubations are not long enough.
But when you know that it's that you get more because it takes time, some are faster and some are not, or because you are over assessing.
Within a reasonably short time, the increasing number of cells is only due, or mostly
okay.
due to labeling.
Okay.
If you label them for a long time, then the other problem comes into play. Okay, so
Okay.
this is not a perfect experiment, but if the lab chief
Is happy. We are happy.
if the lab chief doesn't want an extra accurate measure, but just a reasonable one, that's perfect. If you want a very, very accurate measure, then you have to go to some length. For example, you might use a cytoplasm. Then you ask, do I have the right instruments when designing the experiment? And you say, no, you don't have it. Okay, let's go somewhere, let's collaborate with somebody who has a cytofluorimeter and ask him if he wants to do an experiment with me and help me with mine. Right? Good. The final example I want to show you is no longer a theoretical example, but a real situation in the laboratory. Let's look at this paper published quite some time ago. A PRB-independent mechanism preserves the postmitotic state in terminally differentiated skeletal muscle cells. A long title that basically says, if I have terminal differentiated postmitotic, no longer capable of dividing, skeletal muscle cells, they will remain growth arrested, incapable of dividing, even if I remove PRB. So what keeps them arrested is independent of this gene, of this protein product here called PRB. It is the so-called retinoblastoma protein. All right. What the author knew before doing this study was that PRB is required to initiate the postmitotic state. Because in physiological conditions, that is, among other things, in the presence of PRB, when you remove serum, that is, you remove growth factors from the medium of myoblasts, growing muscle cells, they differentiate into terminally differentiated muscle cells, postmitotic cells. These nuclei here are in black because they no longer incorporate UDR if you add UDR. So the cells are growth arrested. This requires, it was known at the time, that this transition here requires RB because if you take RB minus myoblasts, that is, myoblasts that have don't express RB, knockdown, you do get differentiated cells, but they still keep incorporating UDR. They still keep replicating UDR. In fact, they do some a certain thing called endoreduplication. That is, they replicate and replicate their DNA over and over again until they die. This is why these nuclei are bigger than these ones, because they are endoreduplicating their DNA. All right, so this knowledge, which existed already in the literature, clearly showed that a B is required to establish the post-mitotic state. If you don't have RB, the cells never enter the post-mitotic state. But the question the authors had in mind was, once it is established, the post-mitotic state, is RB still necessary? That's the question. So,
They were asking
Second.
if they want
Give
to.
me a second.
Okay, sorry.
So, what we're going to do now is to go through two of our levels in the scientific process. First of all, the experimental approach. So we'll think about a way to approach the problem, and then we'll do the experiment. Okay? So, how would you assess whether, once you are in this condition, you still need RB to remain growth arrested? That's the question for you, and Samuel.
No, I was asking a question. I don't have an answer.
All right.
Still, I don't. It's like the question they are asking is if we take out RB, what they expect the cells to de-differentiate from that state.
At least to incorporate the UDR again. They might
Okay.
also de-differentiate.
Because yeah, they thought that RB was the one keeping in that state, or at least that one that.
The authors didn't think,
Okay.
they just wanted to know whether it was necessary or not to keep the cells growth arrested.
Can I understand what they do? They incorporate when the nuclei is green, are green.
Bromodeoxyuridine was just
Okay,
said.
bromo. So they are in replication, nest phase.
Yep.
Can we like silence the RB gene like not with a knockout mutant, but maybe with RNA interference? So we do
Yes.
it later and.
We can, but the authors didn't do that because they thought, well, we can interfere with RB, but most certainly some RB protein will be left there because RNA interference is
Yeah.
rarely 100% efficient. So how do we know that there isn't enough RB left to maintain growth arrest even though we have eliminated most of it? So they chose a different way, but this is a viable approach.
A chemical treatment for blocking. Some molecule will attach RB.
Selectively blocking RB, unfortunately, is not possible.
Alright.
If there were a chemical inhibitor, yeah, that would have been a possibility.
Adding the target of the product of RB
That being the target.
to keep a part of RB with our enzyme protein to know how much is in the cell and see.
Okay, so you, I'm not sure I understand. You want to measure the amount of RB
Yeah,
in the cells?
and isolate it, occupate it, adding I don't know, the target.
Oh,
Of
okay.
the-
So basically, you want to titrate RB away from its natural target
Exactly.
by adding an artificial target, a so-called decoy target.
Okay, decoy. Yeah.
Yeah, that would be a possibility. That would also probably not be 100% efficient. And with RB there are difficulties in doing that, but it's a good guess. It's a good thought. What else can we do? Yes.
Maybe using like the Kralox
That's a good idea.
or some
Although you
other.
have been particularly silent today, so you have an idea.
is RB, an enzyme?
No, it's not. It's it's difficult to define. It's a co-transcription factor. It's a transcriptional repressor. It's a regulator of the cell cycle and a tumor suppressor. But biochemically, we don't fully understand what it does.
Do we know if it needs other proteins to work?
It does.
Okay.
But if it did,
We could block the production or the activity of the proteins that it needs.
it might have been a way, yes. Unfortunately, it doesn't need a protein.
We could somehow.
I think I'll stop here because you gave a range of good suggestions. What the authors indeed chose to do was to use the Krylov system. You have to remember that at the time RNA interference did not exist yet. This is 2004, the time of publication.
Okay.
The experiments have been done even before 2004. So they used the RB-LOX system, in which they took cells whose RB is surrounded by two LOX-P sites. LOX-P sites are recognized by a recombinase called CRE, which specifically recognizes them and, in the proper arrangement, cuts away whatever is in between them and recombine them into a single one. So, what the authors did was to take cells in which RB was surrounded by LOX-P, and then they induced differentiation in the presence of RB. So the myotubes became physiologically postmitotic, and then infected them with a recombinant adenovirus expressing CRE. That excised RB, and the result was essentially a 100% excision of RB. This is a PCR showing that exon 19 of RB was present. This was the expected size in control cells infected with an empty virus, and was drastically reduced in size because piece had been cut out when the cells had been exposed to CRE. You see there is no wild type band here, so there is basically no gene left intact. Okay. They also determine, and we are going into the experimental design, because this shows that the gene is no longer there. But that's only the gene. Then you have to consider that once you knock out the gene, the protein is still there,
The leftover.
and it needs to decay completely before you are left without RB. How long does it take? The authors didn't know. But after a certain time, I think it was 2 or 3 days, they took the cells, lysed them, and did a Western blot for RB. Does it make sense? This is what they found. They loaded 30 micrograms of cell lysate from control or CRE-exposed myotubes, and they found that there was a lot of RB in the control myotubes, but no detectable RB in the CRE-exposed ones. Very nice. Except one person in the lab came up and asked, well, how do we know that our Western blot is sensitive enough to detect trace amounts of RB? How do we assess how sensitive is our Western blot?
use less control. I mean, with
I know.
Western with 1 nanogram of control.
Yes? Karim agrees. Do you? Do you? Okay, that's exactly what they did. Instead of loading 30 micrograms, they loaded 1/4 and then 1/4 of it. And they found that even if they reduced the amount of extract 16-fold, they were still able to detect RB. Probably even less than they could see RB even in further dilution. They don't show the data. Even if they had. So basically, their Western blot was sensitive enough to detect RB even if it had decreased 16-fold compared to wild type levels. So they concluded that it was reasonably sensitive. Although no Western blot is indefinitely sensitive. At some point there will be a threshold, there will be a limit of detection, as we call it. Good. So what's the outcome of the experiment? Now we know that we are excising the gene. We know that this results within a certain time in the disappearance of a B, and the result is the myotubes still do not incorporate BUDR. Great. So the answer is PRB is not necessary to maintain the post-mitotic state once it has been established. Except that this is a one-point experiment. Isn't it missing something?
I didn't understand, sorry.
I'm showing you a single picture. In the absence of other information, we can assume that the authors just did this experiment, removed RB and found that the cells did not incorporate BUDR. Is that enough to conclude what they did? That PRB is not necessary to maintain the post-mitotic state. Think of this experiment in an abstract way. What is
You
it?
need
What
a
is it?
control.
You need a control. What kind of control particularly?
Negative. No.
Positive.
This is negative already.
Okay.
And it's pretty negative. The nuclei are pretty dark. So the authors were. But what would be an obvious negative control, which the authors certainly had?
You could use the first mutant line you showed us, the RB minus minus.
No, because
They
those
don't.
myotubes would incorporate BUDR. A negative control, in this case, is something that does not incorporate BUDR. Wild type myotubes, yes. Okay. Certainly the authors said wild type myotubes, even if they don't show it. But then it's the more difficult part. You remember, we said that one important purpose of positive controls is to validate negative results. And this is a negative result. Now this is not just an academic quid ball. If you see that these myotubes do not incorporate UDR, I can easily ask, did you wait long enough? Did you incubate long enough? Did you allow RB to decay long enough? You showed me that 1/16th is not there. How about 1/20th, 1/40th? I don't know. Is it possible that these cells are simply badly damaged by Cree? They're just dying, so they don't incorporate UDR. So you need a positive control. What would that be?
Wild type plus Cree.
Without Cree. Share the mutants with Krolov system, but without the virus with Cree.
That wouldn't be a positive control because that wouldn't incorporate UDR, right?
Yeah. Oh, okay.
So it's not a positive control. I mean, it's something that does incorporate UDR. That's a positive in this case. Because what we are measuring, what we are assessing is UDR incorporation. So we need a positive for that. And so you don't have an answer for that, I'm telling you. Because you don't know what's necessary to know to present a positive control. But in principle, what should a positive control in this experiment be? Abstractly speaking.
What should a positive control do?
You should incorporate UDR, yes. How would you design a positive control? That's the design, the experimental design in this case. What control do you want to make?
Should be space. No.
Well, a positive control is a myotube that incorporates UDR, right? Okay. How would you make a myotube incorporate UDR? Again, I don't want the specifics because you don't know. But a positive control should be any known situation that allows myotube to incorporate UDR in the same conditions as this one. That is, after being exposed to pre, after having its RB delivered. And so what the authors did was to
Vector.
use an adeno vector that expressed an oncogene called E1A, which was known to induce myotubes to synthesize DNA. And so when they took these very same myotubes, actually parallel plane, and after exposing them to pre, they infected them with E1A, they showed that they did incorporate UDR. So it means they weren't damaged, they were capable of synthesizing DNA if placed in the proper conditions. And so this was the positive control that validated the negative results here. These, in the absence of this experiment, of this control, they wouldn't have been able to draw conclusions from this result here, because this result might have been completely artifactual. It might have been due, as I mentioned, to a variety of conditions, including the cells just being dying. This shows that they weren't dying. They were still capable of synthesizing. So, when you are in the presence of negative results, the authors were sort of anticipating that they would be. But anyway, even if you don't know what results to expect, you need to include a positive control. And so once you get a negative result, the positive control allows you to consider the negative result valid and draw conclusions from it. If you don't have this, you don't do anything with this. Is that all right? Okay, so in this case, it was a matter of, first of all, experimental approach. We have to find a way to create a meaningful positive control, and then of experimental design. So first, we expose these myotubes to pre, let them eliminate RB exactly in parallel as this one, and then we do one further infection with UDR and determine whether they can or not incorporate UDR. This is the way the experiment is done.